All right, so another section here, but at least we're moving away from frogs this time. I'm pleased to introduce Megan Obar and McKenna Smith, who were in a class of mine last fall, and we took a project in there, and sort of took a project in this general toxicology class, and we're working with marine animals and marine features, and they and their group really took their project and really enjoyed what they were doing with that and generated a lot of good data and generated a lot of what we're going to learn a little bit about today. Estelle Richards, was an author on the paper on this as well, but she's graduated, but she's here back today, so thank you.
Okay, so our project was on the preliminary developmental toxicity of methylene blue and orange G to grass shrimp embryos. Methylene blue is a common dye used in most biology labs. It's often used to dye bacteria and tissues to better see their shape and structure. And orange G is an organic sodium salt. It's often combined with other yellow dyes in an alcoholic solution to stain erythrocytes in trichrome methods. These are the grass shrimp. They are tiny shrimp that live in marshlands and estuarine waters. They're found along the Atlantic coast and the Gulf Coast. They eat worms and algae and small crustaceans. The gravid females, as you can see in this picture here, they carry the eggs with them until they hatch. And they hatch into free-swimming larvae after about 12 to 20 days. Adulthood is reached after 2 months, and the average lifespan is about a year. We hypothesized that methylene blue would be more developmentally toxic than orange G, and our objectives were to determine the LC50 for methylene blue and orange G, observe the embryos for any abnormalities, and use the larvae length for comparisons of concentrations. To collect the grass shrimp, we took a field trip to Dauphin Island in late September of 2021. They were counted, and 50 shrimp were placed in a gallon-sized Ziploc bag with water and substrate, and then they were put in a cooler and brought back to Jacksonville. Once they were back here, they were put in a large saltwater tank, and the eggs were collected from gravid shrimp once a week and taken directly to lab when applicable, because some weeks there were no gravid shrimp. Eggs from gravid females, days about 5 to 10 age, were obtained and separated individually into well plates, 24 wells per plate. 6 plates were utilized per dye of varying concentrations. Controls held no dye, only salt water at 15 parts per thousand. They were exposed to 2 days hatched, 12 days old at 27 degrees Celsius. 1 egg and 2 milliliters of concentrate solution added to each well, and then placed in an incubator on an oscillator until 2 days post-hatch. Periodically, all the eggs were examined for mortality and hatch status, and the mean, standard error, and probit analysis were done using SysStat.
So here we have the results of our experimentation. These are the graphs of our second experiment that we did with varying concentrations. Initially, for our first experiment, as a way to just test the waters to see how everything would go for our experiment, we had both the methylene blue and the orange G start off with our control, 10 milligrams per liter, 25 milligrams per liter, 50 milligrams per liter, and 100 milligrams per liter in concentrations. Then we moved and did the exact same thing for our next experiment. And as we see from experiment 2, our concentrations for orange G minus the 50 from a little bit of an outlier. Overall, the mortality rates or mortality mean for our embryos were pretty stagnant. No real big change between each one. For our concentrations in methylene blue, however, you can see a steady incline for mortality between our embryos. For our next experiment, since we saw that methylene blue had increased mortality rates regardless, We kept the concentrations for methylene blue the same. However, because of the stagnant mean for the orange G, we increased concentrations from a max of 100 to a max of 250. But as you can see, there was no real change in terms of mortality rates for orange G, but there was an increased mortality rate for methylene blue. We also did length measurements for all of our embryos by extracting one from each well that was still alive and measuring them on a ruler that I will show in the next couple of pictures. And we took all these measurements from both the controls of both dyes, as well as the the next one that had the highest amount of living living samples. So for our orange G, we did our max, which was 250 milligrams per liter, and for our orange G, we did 500 for orange G and 25 for methylene blue. And as you can see, there's no real big difference in lengths. There were no deformities that we could tell from any of our samples. However, in the max concentration one, there is one single outlier, but that's because of how difficult it was to measure this one in particular shrimp. The way that our measurements went is trying to get them into the right position in order for the measurement to be taken through our measurement program. However, it did not work out that well, or as what we wanted for that singular one. We have our standard error link here at the bottom, and as you can see, there's no real big difference between any of them. So no deformities, no difference in length when it concerns these dyes with these shrimp. For our results for the orange G, there was no LC50 reached. We cannot find any big difference between the mortality rates. There were little to no growth deformities, but there did seem to be a discoloration mainly in the respiratory system. As you can see in the picture here, when we were measuring them, there's the ruler at the bottom, but you can also see that there's a discoloration in the upper respiratory system around this area. And what we think that happened there is that while it did not penetrate into the chitin of its outer skeleton, it did in fact intrude during the respiratory system whenever it was breathing in the water. But that just goes to prove that the orange G does not penetrate into the shrimp itself, just through the respiratory system. This is our methylene blue results, where we did get an LC50. So 50% of all of our samples died relatively within a 54 milligram per liter concentration. We have an upper limit of 67.117 and a 44 for our lower limit. While the lower hatching rate increased with increased concentrations, there was no significant deformities for these shrimp as well, but there is staining noted in all deceased and eggs and larvae undergoing tissue decomposition, meaning that while they were alive, no methylene blue integrated into their body, neither from respiration nor from intruding on the chitin itself. So in conclusion, we found that methylene blue had a higher toxicity in the grass shrimp than in orange G. Orange G has little to no effect on the health of the grass shrimp, even though it does cause pink discoloration. The LC50 for methylene blue is, of course, 54 milligrams per liter, while OG has no LC50. And of course, there was no difference in length for either chemical from controls to exposed, as well as any other deformities. So for our acknowledgements, we'd like to thank Dr. Rayburn for this opportunity to conduct this research. We had a really great time just going through the entire process. We'd also like to thank the school itself for providing our supplies as well as our areas for research. And of course, these are also our references that we use for our research as well.
Questions? I'll throw one your way there. Can you go back to your graph graphs where you showed the experiments 2 and 3, I think. There, that's good, that one's there. You got your methylene blue experiment 2 and your methylene blue experiment 3, correct?
Yes.
What is the problem with the experiment 2? Is there a problem with that experiment?
Potentially, we did have some problems with the incubator at the time. We were trying to make sure that it was sealed enough to where our specimen could still breathe, but not get dried out, since that was a huge deal that we had with the first experiment. So it could be potential that the either the eggs themselves were doomed from the start, or it could be that our taping kind of
Suffocated.
Yeah,
I'll
suffocated.
make my question more direct. Why is that blue, you're answering it, why is that blue line on control so high?
that it could just be from
That's what
natural.
you were just saying.
Yeah.
That's what I'm saying.
Yes.
So do you think the grass shrimp will come in contact with these naturally in the wild, or are you doing this more of a grass shrimp like an indicator? Do you understand my question?
Yes, sir. While we don't think that they would necessarily get in contact with these dyes in nature, it is potential that we can expand upon these dyes onto other organisms. They're mainly used for microorganisms looking underneath the microscope, but we don't know what their effects are on other organisms. The shrimp themselves are crustaceans, so we wanted to see the effects on crustaceans themselves before potentially in the future experimenting on other organisms.
Thank you.
Orange G was actually tested with ferrets and a couple of large mammals, and it caused anemia. So it was just a dye that we decided to try on crustaceans.
All right, let's thank our speakers again.